Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. I hope you all are fine and well at your home. Dear students, I welcome you in Pakistan International School Taif Virtual Learning System. This is grade second year's biology class, and today's lecture we are continuing chapter number 23, and in that we will see about chromosomes and DNA, and today's topic is gene expression. We have started gene expression before, and today we will see its next part, that is about transcription. Gene expression means that the DNA makes protein while an intermediate RNA is known as central dogma of molecular genetics. This is also called gene expression. The genes of the DNA just express themselves into phenotype via protein synthesis. So that protein synthesis is having several phases. First phase we have discussed before, that is transcription. That is replication we have seen before, and in today's topic we will talk about a transcription. So gene expression is, we can say, changes in DNA may change the resulting protein, but changes in protein cannot feedback and change the DNA. So genes express themselves in the form of protein synthesis, and which is irreversible. So the process by which a gene information is converted into structures and functions of a cell by a process of producing a biological functional molecule of either protein or RNA. That is, RNA is a gene product that is taking the messages from the DNA. So, gene expression is actually assumed to be controlled at various points in the sequence, leading to protein synthesis that is not a, we can say that, unattended process. So the first of all, from in the gene expression, the first very important stage is transcription. And transcription, you can see here, DNA transcribe RNA, and RNA then translate the messages of DNA into amino acids, and then amino acids are getting folded into the protein, a required protein. So protein synthesis, that is also called central dogma. So that is done in four stages. First is transcription, then RNA processing, that we can say that post transcriptional processing, and then there is translation, and then we are having post translational processing. So let's talk about a transcription first. So transcription means actually synthesis of an RNA that is complementary to one of the strands of DNA. You can see here in this diagram that here RNA strand is going to be transcribed from the DNA strand. So just this, you are just you can say that this is a simple process, but actually this is complicated process, which we will talk about in detail right now. Now we will say different phases of transcription, because transcription is not occurred at once, it is having several phases. First is initiation phase, then elongation phase, and then termination phase. We will talk about all of them in detail one by one. There are certain there are enzymes which are required for transcription, but like replication, it does not need so many enzymes. Just one enzyme is needed, that is called RNA polymerase. And that is the enzyme that controls the transcription and search that also search DNA for initiation site. Then it unwinds a short stretch of double helical DNA to produce a single stranded DNA template. Just this only enzyme acts for all of the processes. So it selects also the correct ribonucleotide and catalyzes the formation of phosphodiester bond. Then it also detects termination signals where transcript ends. It means from initiation up to the termination, including elongation, just polymerase, RNA polymerase is required. So it performs varieties of functions. If we see the structure of RNA polymerase, so the structure of RNA polymerase, you can see there are several units, and that that is having four subunits. That is alpha, two alpha subunits, then one beta and one beta prime subunit is there. One more subunit is present that is called sigma factor. Sigma factor is required when it is going to attach to the 5 prime end of the DNA. So without sigma factor, it is called core enzyme. So it is required for polymerization activity. But with sigma factor, it is said to be hollow enzyme and it requires for correct initiation of transcription. So it means it is required for binding to the promoter. When it binds with the promoters and the sigma gets off again, and these all four subunits, they are required for further elongation and polymerization. So that is about eukaryotic. And prokaryotic RNA polymerase, that is all both of are having almost the same one. So you can see alpha, these two are alpha. It needs for chain initiation and interaction with the regulatory proteins. Oh, actually these are the functions of these subunits which are present here. So beta requires for chain initiation and elongation, beta prime requires for DNA binding, and sigma requires for promoter recognition. So all are having different functions in the polymerization reaction. Then we will see not only one RNA polymerase is present, but there are three types of RNA polymerases. Number one, RNA polymerase 1, that is located in nucleolus and that transcribes ribosomal RNA, because for protein synthesis we require three types of RNAs. So polymerase 2, that is localized in a nucleus and it transcribes messenger RNA, and here polymerase 2 is required for a transcription or carrying the messages of DNA. And more smaller nuclear RNAs, snRNAs. Now polymerase 3, that is localized in nucleus again, and actually it is having the nucleolar nucleoplasm interface, like it functions for both nucleoplasm and nucleolus, as well as it transcribes transfer RNA and other small RNAs. Let's talk about the first phase of this transcription, that is initiation phase. So in that phase, to start or initiate the RNA transcription, RNA polymerase, that is said to be RNAP, recognizes and binds to a specific region in the DNA that is called promoter region. So let's talk about something about promoter regions. So in eukaryotic promoter region, promoter lies upstream of the gene, like where the gene is present, upside of that one. And there are several different types of promoters found in human genome. So if we talk about the promoters names, and then we are having their sequences. So you can see here there is cat box, Tata box, GC box, and cap site also. Actually, these are the sequences in which you can see here cat comes CAAT, and here is TATAA Tata, and GC and G, and tag, that is the cap site of that promoter region. So these are the different sites which are included in the promoter regions, where these sites, where the sequences are found. So the RNA polymerase just attach over that. It recognizes with the help of sigma a factor, or sigma subunit in the polymerase. It just recognizes the initiation site, that is called promoter site. So that very important is there, that there is a Pribnow box, that is also called Tata box, that is consisting of 6 nucleotides, and it is located to the left side of 10 bases upstream from the starting point of transcription. You can understand what, like in this diagram. If we talk about prokaryotic promoter, so you can see here promoter. It's this is the site from where it will start. It's a transcription towards 3 prime end. So from here, that is -10 sequence, that is called Tata box, and here is -35 at number 35 towards the downstream, that is -35 sequence. So -35 box and -10 box, they are called Pribnow boxes, and they are consensus sequences. So the two boxes are far apart from specific distance. They are located at the same face of the double helix, like at the coding strand. So when from this starting point, the RNA polymerase just identify this and this sequence and just attach over it to start the transcription. So eukaryotic promoter region, so you can see here, there is a from almost it start. This is the region where the transcription will start. The site is present, that is +1, and mm, up to -25, that is Tata box, and that is on the coding side. And then at -50 and -100, that is up to -100. From -50, -70, that can be. There are some boxes, GC boxes and cat boxes are present here at the yellow region. So these are the some sites, if they are present consecutively in some places, so that is the promoter region, and RNA polymerases attach over it. So two parts of promoter, that's a core promoter and basal promoter, upstream promoter also. So core promoter is Tata box, and transcriptional site starts from Tata box, and initiation complex binds to this core promoter. Here that initiation complex, that is of polymerase, RNA polymerase, that binds over there. So upstream elements are responsible for the regulation of the transcription, that is all these regions. So in initiation phase, polymerase binding causes the unwinding of the double helix, and at least 12 base pairs, not more than this, 10 to 12 base pairs, and this is followed by RNA synthesis at the starting point. So RNA polymerase starts binding to RNA chain. It assembles nucleotides, that is triphosphate, adenosine triphosphate, guanosine, and cytosine, and uracil, because in RNA thymine is replaced by uracil. So after first nucleotide is in the place, polymerase joins the second nucleotide to the first and then forming the chain, and chain initiates. Now we will talk about elongation phase. So RNA polymerase directs the sequential binding of ribonucleotides to the growing RNA chain to 5 to 3 prime end. So you can see here in this one, this is 5 prime and this is 3 prime end. Very important thing is that the this is RNA polymerase. This is RNA polymerase. It has been started from here, but now it reaches here. So the first strand is coding strand and the other one is template strand. Actually where it unwinds, just it recognizes the 5 to 3 prime end. So when this is 5 to 3 prime end, but it has to be formed in opposite direction from here to this side, from right to left side. So this 5 to 3 prime end, it will elongate the chain and attach the nucleotides here towards left side. And this 5 to 3 prime end, that sequence is very important. So this 5 to 3 prime end, when it combines here, or it makes the RNA, so it is the exact copy of this coding strand, means it codes for RNA. So here we can say the 5 to 3 prime end, and then guanosine in DNA leads to cytosine, cytosine to guanine, thymine to adenine, but thymine in DNA is replaced by uracil in RNA. Adenine in DNA shows up for uracil in RNA. So now here you can see that a template strand, this one is template strand and this one is coding strand. So messenger RNA is formed in 5 to 3 prime end, and when it is forming, like this is coding strand, you can see here 5 to 3 prime end. Now this one is like this one is messenger RNA, transcribed messenger RNA, because you can find here uracil, and this is the DNA template. So when you see this thymine is complementary to adenine, adenine to uracil, guanine to cytosine, and guanine to cytosine, and adenine to uracil. So you can see this strand, transcribed strand, that is exact of this coding strand. So it means, except this thymine and uracil, it means this messenger RNA is the copy of this coding strand. Actually, it's try to copy this message. So the same, but there is a difference because in RNA there is uracil in place of thymine. So the same of this coding strand. Elongation step of transcription, you can see here. Here is unwinding. Here is unwinding and RNA polymerase just forming this RNA and TPs, like these are nucleotides just are placing here. This is the template and this is coding strand. This whole is RNA polymerase. This RNA polymerase moves to this way, polymerase movement, it moves and then unwinds, and at the back, from where RNA transcribed, it just rewinds over there. This can also, you can see here the process also, how this is RNA polymerase that transcribes. So template strand, and these are TPs that are like nucleotides that are just attaching with that. Phosphodiester bonds is forming, as well as that is transcribed strand is moving out of this polymerase. Now you'll see the termination phase, that is also very important. That is also called signal termination. because this works in conjunction with RNA polymerase to loosen the association between RNA and DNA template. So RNA dissociate from RNA polymerase and DNA and so stop transcription. So the product is immature RNA and pre-mRNA is formed. In termination phase, very important things are there, that in this mechanism transcription is terminated due to specific sequence is terminated DNA. That specific sequence, the terminated DNA, the terminator DNA contains invert repeat which cause complementary pairing as transcript RNA form hairpin structure. So this invert repeat is followed by the larger number of TT, almost 8 base pairs on the template. So you can see here, this is the termination phase, that is called poly-U site. So before that, that forms this hairpin loop like structure where there are lots of GC rich area. So when the GC rich area, they are just they find their way, or G just forms the bond with C and C with form a bond with a G, because both are complementary to one another. So they form bonds here, and here the hairpin loop like structure is formed, and at the end this is the way, like this is the starting point, this is the ending point. At the end, it ends over there where the poly U tail is formed. So in the DNA, poly A will be attached with poly U, and here then after that no transcription will occur, and this is the last point, like the termination sites. So the primary product of RNA transcription, the HNRAs, like we can say the high nucleic RNAs contain both intronic and exonic sequences, so that have to be removed, and there are some other changes during this process. So that is called post-transcriptional modification. So in post-transcriptional modification, that is the process in which precursor messenger RNA is converted into mature messenger RNA. For that, three types of modification has to be applied. Like at 5 prime there is capping, at 3 prime there is polyadenylation, and RNA splicing is there. So in 5 prime capping, like at the 5 prime, it is in the nucleus before going out into the cytoplasm. What happened at the 5 and 7-methyl guanosine cap to 5 prime, and the first step of post mRNA processing. So it occurs co-transcriptionally after the growing strand has reached 30 nucleotides. So after that it is done. So at 3 prime end, polyadenylation, like poly A tail is in the primary transcript that is added at 3 prime end. Now the RNA is safe because lots of enzymes in the cytoplasm are present to break down messenger RNA. To save them, it has to have a cap which is at 5 prime end and have a tail of polyadenylation, because there no bond will be formed after that, and no enzyme can work over it. So RNA splicing is also the process applied over there, like introns are removed from the RNA. Like these are some extra sequences which are going to be formed, which are not needed during protein formation. So introns are removed by enzymes, and remaining exons connected by single continuation molecules, and that splicing reaction is catalyzed by large protein complex that is called spliceosome. That spliceosome catalyzes all this process. So you can see here post transcriptional modification. This is the transcribed RNA, and addition of 5-methyl guanine, a cap is applied. Here are intron and exons. So what happened? Introns are discarded and removed by spliceosomes, and later these exons are just like combined and attached with one another to form a single chain of that messenger RNA, and at the end polyadenine like adenylation is done, and this tail is like formed at the end or connected with that messenger RNA at 3 prime end. Here at 5 prime end, that is addition of a cap. So now it is ready to move out of the nucleus in the cytoplasm for protein synthesis, the very important process. It will attach to ribosomes for translation, translating of their its messages. So that's it for today, dear students. I hope you understood this lecture, and very interesting. We will talk about its next step, that is translation, that is actually the protein synthesis, in the next lecture, inshallah. Wish you all the best. Allah Hafiz.